Hello. This lecture is on the research that we do in developmental psychology. So with research, our core question is, how do we know our behavioral explanations are accurate? So remember those behavioral explanations or theories that we talked about last time. How do we actually know that they're good theories? And that is through research. So we're going to learn about the scientific method, comparing the different research methods. They each have their own advantages and disadvantages. We'll also talk about the research involving time spans, as well as ways to conduct research in an ethical manner. So, how do researchers gain supporting evidence for their ideas? Why do theories get revised or thrown out? Research. So research is conducted using the scientific method. It is a set of assumptions, rules, and procedures that scientists use to conduct research. So we typically start out with a research question, something we're curious about. We develop a prediction or a hypothesis about why we think this observed phenomenon is occurring. And then we develop a way to test it. So picking out a research design and methods of data collection, we gather that data, we analyze that data, and infer conclusions from it. And the type of research design that we choose influences or, well, determines really what type of conclusion we can draw, whether that be just quantitative data, whether that be a correlational relationship or a causal relationship. Next, often research results are shared, typically through publication in a peer-reviewed academic journal. This allows for others to replicate your research or to study new questions that your research has raised. And that's why the scientific method is circular, right? New questions arise as we go along. Before conducting research, or before conducting a study, researchers need to decide which method of data collection they'll use and what research design or overall plan that they'll use to test their hypothesis. So some of our terms that I've mentioned here, hypothesis, that is a prediction drawn from a theory. So it shouldn't be a wild guess. It should be in line with what previous research has found, or at least doesn't contradict those observations that have been conducted. The research design, the method for collecting, analyzing, and interpreting data, I tend to think of this as the overall game plan. Some other important terms, population. Population refers to the entire group of interest that you want to study. The sample is a subset, so a smaller group that we get from the population through random sampling. So, I mean, we don't always get our samples that way, but that's the best way to get them, which is if we actually contact everyone in the population and give them the opportunity to participate, or well, not everyone, but we randomly select people from the population to invite to join the research. It's important that our sample be representative of the population as opposed to bias. So what this means is that our sample reflects the characteristics of the population at large. If I'm interested in college students, for example, my population of interest, college students, my sample should be college students, right? I want a good size sample, large sample, so that I'm more likely to get similar demographic information in my sample compared to that in the population. So for example, if the population of college students is in the United States is, and I don't know this for sure, but say it's 60% female and 40% male, I want my sample to also be 60% female and 40% male. And so I want those same demographics, gender, sex, race, ethnicity, age, all the variables that could be involved. I want those to be reflected in my sample so that when I get my results and draw my conclusions, I can say these results, these conclusions don't just apply to my sample, they apply to the entire population because my sample represents that population accurately. So a good understanding of research is key to deciding which type to use, what types of conclusions can be drawn from results. It's also useful for evaluating the research of others, right? Being a competent consumer of information. There's a lot of misinformation out there, and news outlets often misrepresent findings such as inferring causation from correlational research results, for one. And you know, they do that to sell, you know, to get clicks, to it's not necessarily done. I mean, it can be done on purpose, but I hope it's not done on purpose, but more just as a way to try and draw people in. So let's talk about our data collection methods. Depending on what type of data you want to collect and what research design you're using, you must choose what data collection method or methods to use. I have a list of common ones here on my slide. Each of these methods is appropriate for certain situations and inappropriate in others. So I'll go into these in more detail. The first, the case study. The goal with a case study is to gather a full in-depth picture of an individual's or sometimes a small group's psychological functioning. It typically combines information from a couple different methods of data collection. So it's usually using interviews, observations, test scores, diary entries, whatever way they're kind of gathering data this way. They're very well suited for studying the development of individuals who are rare and who may vary widely in characteristics. Child prodigies are one such example. So, I mean, we don't run into a lot of children who are math prodigies, but it might be useful to learn more about them and, you know, why that occurs. The big strengths, you know, they're giving you a lot of information in depth, and they can lead to further, more refined research. The downside or weaknesses of this type of method is that information is often collected unsystematically, more subjectively, and that leaves researchers' theoretical preferences to bias their observations and interpretations at times. The other really big limitation of this method is that because only one or a couple people are being studied, their results are not generalizable to the population at large. With systematic observations, researchers are observing and recording the behaviors of research participants. And this can be done either in a natural setting or in a controlled setting. So this is where we get naturalistic observations and laboratory or controlled observations. With naturalistic observations, you're going into the field, the natural environment, to record the behavior of interest. And it doesn't necessarily have to mean in like nature. If you're interested in studying college students, say, the natural environment might very well be a classroom. So again, just a natural environment. The big strength of naturalistic observations, they're more likely to reflect everyday life because individuals feel comfortable, more comfortable than if they were in a more controlled setting. The downside, not all individuals have the same opportunity to display a particular behavior. So if you're interested in studying sharing among children, for example, and you go to a child care center and watch to see if a child shares, but maybe no other child asks that child for a toy, then you can't really record any meaningful data. Laboratory observations, on the other hand, because they're in a controlled environment, I can ensure that all of my participants have the opportunity to display that behavior of interest. I could have a room set up with toys and the child could be playing, and I could, send a research assistant in to ask, hey, can I play with that toy, to see if the child shares, right? So that's the big strength there. Gives us a bit more control over the situation. The downside of structured or laboratory observations, they may not reflect real life. People are more likely to change how they behave when they know they're being watched, and it's definitely very apparent that that's the case in those settings. Self-reports. Self-reports, there's a lot of different types. So I have clinical interviews, and then I have structured interviews, surveys, and tests or questionnaires. And I separate out the clinical interviews from the other type because they have specific characteristics, whereas the others are very similar. So the big strength of self-report methods in general, we are asking participants to report on their own behaviors. The big downside, and you'll see this on both sides on the slide, is that participants may not be accurate in their reports. So they might just not remember the information, they might forget, it might not be something they think about. They could also potentially lie to make themselves look better. We call that social desirability bias. So I mean, self-reports, sometimes we have to rely on those because we can't accurately measure things like thoughts and feelings using observations. But with the self-reports, you're opening yourself up to that inaccuracy in the data. So again, pros and cons to all of these methods, and you have to weigh those when making decisions about how to conduct your research. So the clinical interviews, this is where researchers ask questions in a flexible conversational style to probe for the participant's viewpoint. The big strength of this type of data collection is that it allows for the exact expression of a participant's feelings and perceptions, and you get a relatively large amount of information in a brief period of time. The downside, as I mentioned, for all self-reports, potential inaccuracy. With clinical interviews in particular, you are relying on the verbal ability of your participants. So if they're not as good at verbalizing their thoughts, or if they're just tend to be more quiet, you're not going to get as much information than participants who are better speakers, essentially. Another downside, because you are asking, follow-up questions, people can really answer in a variety of ways. It can be difficult to compare the data that you collect with a clinical interview. So for example, if I were interested in studying childhood, whether people had a healthy childhood, a good childhood versus a less, a more turbulent childhood, I could ask an open-ended question like, tell me about your childhood. And who knows what one person's going to say versus another. Some might say, well, I had a great childhood. Some might say, my parents got divorced when I was a kid, and that, was really hard. Who knows? Kind of difficult to compare person to person. With the structured interviews or a survey, a questionnaire, a test, we're using the same questions asked in the same way to all the participants, and that makes it a lot easier to compare their responses. So instead of saying something like, tell me about your childhood, I could say, would you rate your childhood as happy on a scale of 1 to 10? Did your parents get divorced? Did they fight a lot? How many friends did you have? Right? And questions that are close-ended, limited choices in responses, and much easier to compare. It's also a lot more efficient to use structured interviews or surveys or tests. You can use them, with a large group and collect data pretty quickly. The limitations, you're getting less depth in your information. A lot of researchers are combining the two approaches. They try to get the best of both worlds. Psychophysiological assessments. So these data measures are looking at bodily processes like our heart rate, our blood pressure, hormone levels, brain activity, and then comparing that data to the behavior a participant is engaging in. And this type of data collection is very common to the field of cognitive neuroscience. Particularly the brain scan, I should say. And I have a couple images here of an EEG, an electroencephalogram, a CT scan, computerized tomography scan, and a functional MRI, functional magnetic resonance imaging scan, just so you can kind of see the differences in those scans. The big pro of using these types of measures, they're much more objective, meaning our emotions, our point of view, our biases is not going to get in the way. They're pretty clear cut, as opposed to being very open to interpretation. They tend to be rather detailed. The big downside, especially with brain scan methods, less so for things like heart rate, but for the brain scan methods in particular, they are expensive. Most people do not have the equipment or the training to be able to use like a CT machine, right? Lastly, secondary or content analysis. This analyzes data, documents, or other information that's already been collected in order to uncover attitudes or preferences. Many data sets are available. So things like census data, the Minnesota Twin Family Study is a huge collection of data from twins. These can be very helpful in that you don't need to recruit participants for your study or even collect the data. It's already been collected. You're just analyzing it in regards to what you're studying. So the potential issue here is there data that already exists that's even appropriate for your study, and making sure that data is of good quality. Moving to our general research designs. General research designs, like data collection methods, have their time and place, right? They all have their pros and cons. Your research design is useful for determining different types of conclusions. And we'll talk about kind of the ins and outs on the following slides, but our major general research designs are descriptive, correlational, and experimental. So with descriptive research designs, they describe what is going on at a particular point in time. We'll define a research question and a variable of interest. So if you're interested in studying grades or aggression or what have you, whatever variable you're interested in diving into. They don't make a hypothesis and they do not manipulate or control a variable with this type of study. It's really more a time to gather data and to see what is happening. And this is very useful for if we don't know a lot about a particular subject, it's a new area. It can be helpful for finding patterns and to hopefully lead us to more refined research where we're actually going to be able to draw a prediction and potentially manipulate variables. So being able to run like a correlational or an experimental design. The common methods of data collection that we use with descriptive designs, things like case studies, naturalistic observation, potentially surveys, interviews, those qualitative sorts of research. The major downsides or limitations for this design is that we're not testing a hypothesis, so we can't infer cause and effect or correlation or any type of real solid relationships between the variables. We can just kind of report basic numbers. Correlational designs are when we are trying to find a relationship between two or more variables, often for the purpose of allowing us to make a prediction of future events from current information. So if I know two variables are correlated, and if I know what type of correlation they have, positive, negative, or how strongly they correlate, that's going to let me make a prediction if I know the data of one of my variables to predict the other one. And in fact, sometimes we call the variables in a correlational design a predictor variable and an outcome variable. So with correlational designs, you will define your research question. You'll typically state a hypothesis, but you're not going to control or manipulate variables. You're not going to alter any experiences of the participants. Common data collection methods that are used with correlational designs, surveys are very common, naturalistic observations, interviews. They're very helpful for determining if there's a systematic relationship between variables, the strength and direction of that relationships. They're helpful for situations in which variables can't be manipulated because it's unfeasible or unethical. The big downside, though, we can't determine cause and effect. So correlation is necessary, but is not sufficient to determine causation. So we'll dive into that a little bit more here. So researchers who use correlational studies examine the strength of a linear relationship between two variables using the Pearson correlation coefficient, or the R value. A correlation coefficient is a number that describes how two variables are associated with one another. A correlation coefficient ranges from -1 to +1. +1 indicates a perfect positive relationship between two variables, while -1 indicates a perfect negative relationship. The closer the correlation coefficient is to 1, the stronger the relationship is between those two variables. So if you're getting a correlation of like 0.75, that's going to be pretty strong. If you're, the closer you get to 1. The closer you are to 0 is the weaker the relationship is between those variables, to 0, which is no relationship between the variables. So let's do a little example here. Pull up my whiteboard. If I can. Where are you, whiteboard? Okay. So, if I have a perfect positive correlation, a graph of that data could look something like this, in that all the points would align oh so prettily. We don't see that too often in the real world. What we'd actually see is something more along the lines of data points being a bit more spread out. So let's talk about something that is positively correlated, right? Positive correlation. What does a positive correlation mean? It just means that the variables vary in the same direction. So they're either both high or they are both low. And I like to think of positive correlations, positive correlation swing. So kind of like the chains on a swing, when you're swinging, they're both going up or they're both going down. So here, say I'm interested in studying the variables of hours of studying per week and how that might predict GPA. And I'm curious, how many hours are people spending studying? What's their GPA? 'Cause I think there's a relationship, there's a connection there. So say I send out a survey and I ask students, hey, about how many hours do you spend a week studying and what is your GPA? And I get data back, say, this little data point that's going to represent somebody who's studying about 18 hours a day and has a 3.9 GPA. I get somebody else, they're studying 10 hours a week and they have a 3.5 GPA. And then I kind of just get people, all over the place, all these little data points. And in real life, we would, of course, take those data points and plug them into some statistical software that will calculate that Pearson correlation coefficient, that R value for us. And what it's essentially doing is calculating linear regression. So it'll calculate a line of best fit and how much variance really is between all of those data points and that line of best fit. And it will spit out our correlation coefficient of say, and again, I would actually calculate this for the purposes of this example and how I don't actually have numbers for all those little points I drew. I'm gonna just make it up. I will say it's a 0.64. Okay, so it's not a particularly steep line, 0.64, a little midway between 0 and 1. So that's about a moderate strength relationship, meaning these variables, they're related, not super strong, but better than weak. The positive correlation, meaning they vary in the same direction. So most people, they are, for most people, it's true that if you're studying more, your GPA tends to be higher. And if you're not studying as much, you tend to have a lower GPA. In reality, right, we also probably have outliers, someone who studies very little and has a great GPA, or someone who studies a lot and doesn't have as good of a GPA. I mean, that's partly why you'll never see that perfect correlation, right? Those outliers there. What about a negative correlation? So the way a negative correlation looks, I'll show you a perfect negative one. Again, those data points just align oh so nicely. Oops, I didn't quite get them all there, but that's what happens when you try and draw on a tablet screen. So that's what a perfect negative correlation would look like in terms of a real world example. Negative correlations Are negative. Negative correlations mean our variables vary in different directions. So when one variable is high, the other is low, and vice versa. And we can remember this with seesaw. So when one end of the seesaw goes up, the other is down. So real world example, self-esteem and depression. And again, if I were to send out a survey to conduct a correlational study, because I suspect there's a correlation between self-esteem and depression, I could have people fill it out. Let's just say a scale of 1 to 10, right? Where do you fall in terms of your self-esteem level and in terms of feeling depressed? And say I get back, some data from people. Someone says, I have really high self-esteem. I don't really feel depressed. Their data point's going to be about there. Somebody else is like, oh, decent self-esteem. Sometimes I feel depressed. Their data point's there. And someone who's, feeling like, yeah, I struggle with depression and my self-esteem's not great. Right? And all of the other data points that I get from those surveys, plug them into my statistical software to calculate that line of best fit and my correlation coefficient. And you'll notice this is a steeper line that I did intentionally to show it is a stronger correlation. It is closer to that perfect correlation. And say this is negative 0.82. So with strength of correlation, remember, it's only based on the number, not whether it's positive or negative. So between these two examples, self-esteem and depression are more strongly correlated, more strongly linked than hours of studying in GPA. And I don't remember the exact correlations for these, but that is actually true in real life. What something with no relationship would look like, an R of 0, that would be something like GPA and eye color from light to dark. What are you going to get? You're going to get data points all over the place on our scatter plot here. Because, people with dark eyes and light eyes are going to have, high and low and middle of the road GPA. It's just all over the place. So you're not going to get a relationship there. So even if I get that perfect, imagine you get that perfect positive correlation or a perfect negative correlation, is that enough to say there's cause and effect? No, you still can't determine cause and effect. And why not? Well, let's go back to PowerPoint. So the reason why we can't determine causation with a correlation, we'll say the third variable problem, also the reversibility problem. So we don't know if variable A is causing variable B. So for example, I don't know if low self-esteem causes depression. It might. It could be the reverse, though. It could be depression causing low self-esteem. I don't know, right? That's what these question marks are for. Maybe that could be the case. Right, the correlational study is not going to tell us. Then there's also the third variable problem, where it could be that some unknown third variable is causing both low self-esteem and depression, such as maybe a bad breakup. And they call it third variable, but it could be multiple variable, frankly, causing them. So we can never determine cause and effect because we leave ourselves too open to other variables influencing the variables we're studying. If we want to determine cause and effect, we have to conduct an experiment. So with an experimental design, This is where we are manipulating one or more variables in order to see their effect on another variable. We designate these variables, the independent and the dependent variable. You can also have more than two, but so we have a research question, we'll state a hypothesis or a prediction. We'll also control the environment, manipulate at least one of those variables, the independent variable. We don't manipulate, we only measure the dependent variable. And we'll use several techniques to control for other variables to avoid having what we call confounding variables influence our variables we're trying to measure. We'll do that through things like random assignment to groups, which I'll get to in a minute. The common data collection methods we use with experimental designs, laboratory observation, psychophysiological assessments, interviews, questionnaires, things like that. The big strengths, we can determine cause and effect relationships when we run an experiment. The downside, the findings are contained in our findings are conducted or found in highly controlled environments. And so that might not apply. They may not apply to the real world because real world conditions, right, are much more variable and things, you know, those outside variables that we were trying to protect us from are now at play. We also can't always use experimental studies because it might be unfeasible or unethical to manipulate a variable. So things like if you're interested in studying the effects of poverty on children's reading ability, we can't randomly assign students, children to grow up in poverty and some not. That would be unethical, also unfeasible. I don't know how you'd even manage to do that, even if you were so inclined. Unethical, there's no way a review board is going to be like, sure, you can force some children to grow up in poverty. I mean, again, right? Wrong, even if you could do that. If you could, you shouldn't, right? So can't always do a study to get a cause and effect relationship. Sometimes we have to settle for a correlational study, a correlational relationship. So I mentioned independent and dependent variables. Independent variables manipulated by the researcher. They're expected to cause a change in the dependent variable. And our independent variable sometimes is also referred to as a treatment variable because oftentimes we're testing out a treatment, not always, but frequently enough that it's sometimes referred to that way. Our dependent variable is what we're measuring. So we're not making any direct changes to this. We are just collecting that data. And it's expected to be influenced by our independent variable. So for example, if I'm a researcher studying the impact of study time on GPA, and I, assign some students to study for 1 hour and some for 3 hours and some no hours, right? What's our independent variable? Study time, right? We'd have different levels, 0 hours, 1 hour, 3 hours, whatever I said, I don't even remember. And then our dependent variable would be the test scores, right? Checking to see, or GPA, right? How are those study times impacting that variable I'm interested in measuring? Sorry, getting ahead of myself. So let's go do an example. Okay, so say I am interested in studying a new medication called Smiley's. It's an antidepressant. Helps make you smile, right? You can actually see drawing here. So it's going to be my independent variable because that is what I will manipulate or control. My dependent variable will be depression levels, right? I want to see if Smiley's causes a change change in depression level, hopefully. My population of interest, that will be people with depression. Right? That's who I'm interested in studying and testing my medication on. It's not going to tell me much if I give it to people without depression. For my sample, because I can't study all people with depression, I'm going to have to collect a smaller subset. So say I randomly send out flyers to a bunch of people with depression within my state. I might have to change my population to people with depression in my state, right? To make it more accurate. You know, sometimes we'll use a convenient sample. It's people often in our geographic region, and that should be stated in your study results in any publication you make. So I randomly sample, let's pretend I could, right? Randomly sample people with depression, get 1,000 people to be in my study. I then need to take my sample and divide them into the group that will get my new medication, what we call the experimental group, and the control group, the group that we treat exactly the same, but they do not get the independent variable. Okay. How do I decide who goes into what group? In order to avoid bias, I use random assignment. And random assignment, we'll use like a random number generator. This is an unbiased procedure used to assign participants the different treatment conditions. It increases the chance that characteristics like gender, age, severity of depression level, other variables like that will be equally distributed across conditions or across groups. This means that I should have, given a large enough sample and using random assignment, I should have essentially equal groups. So I'll end up with 500 people with depression in each group. I will give them a questionnaire measuring their level of depression. So I have, you know, their baseline data. And then over the next 3 months, the people in my experimental group, I'll give them 100 milligrams of Smileys per day. My control group, they're getting 0 milligrams of Smileys per day. In fact, I'll probably give them a placebo pill. So a placebo is a pill with no active ingredients. The whole idea being that I want to avoid having the confounding variable of expectations from my participants. So participant expectations can change the dependent variable. So if my participants know, ooh, I'm getting a pill that should help me, I feel better already, versus someone who's like, well, I'm not getting anything. I must be in the control group. Great. This seems helpful, right? Maybe they even feel more depressed because of that. I mean, seems likely. So in order to avoid that, we run what we call a single blind design by giving a placebo to the control group. So they are blind to which condition they are in. They don't know, well, and not just the control group, the experimental group doesn't know either. So all of my participants don't know which group they're a part of. We call that single blind. Double blind studies, which is the even better way to do it, avoids researcher expectation effects as well. So the researchers that directly interact with the participants also don't know which group they're in. So they don't accidentally, I don't know, smile more or are kinder or chat more with one of the groups, which could throw off my results as well. So We run the study, say for 3 months, experimental group getting that 100 milligrams of Smileys a day, control group getting 0 milligrams. They're just getting a placebo. Essentially, it's like a sugar pill, like a Tic Tac, except it doesn't freshen your breath, right? And then I take their, I give them the questionnaire again, measuring their depression levels. And if my experimental group has lower depression levels on average than my control group, then I'll know that Smileys, in fact, caused that change to happen. Okay. And I know it caused it because these groups were essentially the same at the beginning. And the one difference between them is one got my independent variable. So that's why we can determine cause and effect with experimental studies. Let me switch. Slideshow. So time span research design. Developmental research designs they're also called developmental research designs, and they are specific types of research designs that are especially suited to studying human development, which requires information for different age groups. We have our cross-sectional design, where participants of different ages are studied at the same point in time. So if I'm studying kids in 4th, 8th, and 12th grade, for example, regarding their relationship with their parents. Longitudinal study, same participants studied repeatedly at different ages. So that would be if I follow a group of 4th graders, interview them about their relationship with their parents, and then when they're in 8th grade, I interview them again about their relationship, and then when they hit 12th grade, I interview them again. Sequential research designs we're essentially running a sequence of similar studies or the same study. So I'm studying participants of different ages over time. So how we do this, we'd have 4th graders, 8th graders, 12th graders, say. And I studied them all, say year 2000. Make this easier with what I'm doing here. Gather their data from each of those groups. And then in 2004, my 4th graders are now 8th graders, my 8th graders are now 12th graders, and I get a new group of 4th graders. My 12th graders from before have aged out of my study. So I'm no longer using them or collecting data from them. 2008, 4 more years down the road, I need a new group of 4th graders. My 4th graders from last time are now 8th graders. My original 4th graders who are now in 12th grade. And again, I'm getting a group aging out. So kind of like a sequence of longitudinal studies, essentially. There are advantages and disadvantages, and I realize these are slightly different order compared to the previous slide. But with longitudinal studies, big advantages, we're getting that change within individuals over time, so we can analyze their developmental process. The downside, they're expensive, they take a long time, years, decades even. And this leads to higher rates of participant attrition. Attrition meaning dropout. There's also the possibility of practice effects. So are they actually changing? And I mean, this wouldn't apply really with the relationships with parents, but if I were giving them like a motor skills test or an intelligence test, right? Are they getting better because they're older, or are they just getting better because they've done this before? Additionally, we can't examine cohort effects. So remember, cohort refers to generation. So because with longitudinal, I'm following one group over time, they're the same cohort. I don't know if other cohorts might be different than them. So maybe their results don't generalize to other people. Cross-sectional designs. So that's the different age groups studied at one point in time. That gives us information on age-related change. but we're not going to get changes over time. So we're not going to get the individual developmental analysis. We can't examine cohort effects because we're only getting information, one-off. Maybe things are different due to different ages. Maybe they're different due to cohort effects. I can't say for sure with this study. Sequential, on the other hand, we're kind of getting the best of both worlds in terms of the advantages. We're getting that change over time. We're able to see changes between participants of different ages. We can study cohort effects with this type of study. The downsides, though, can be expensive, can have practice effects. So let's talk about ethics. So before you conduct a research study, before you gather any data, you have to submit your proposal to an institutional review board, an IRB, institutional review board, and they are made up of experts in your field, experts in other fields, as well as members of the surrounding community who have no stake in this game, if you will. And they will examine your research for whether it's ethical, whether it's meeting these guidelines or not, and will determine whether you get to move forward. So the big ideas with ethical research is to protect participants from harm and from exploitation. So we have the general rule of you don't want to introduce any harm to the participants physically or psychologically, other than maybe slight distress that they might encounter in their everyday lives. You also have to have informed consent, meaning you have to explain to your participants all aspects of the research that might affect their willingness to participate. So any known risks, if that's an aspect there, they have to, they're signing a legal agreement. It has to be in language appropriate to their level of understanding. If they are not capable of giving consent, you have to get informed consent from a parent or guardian, and then you get assent from the individual, whether that be a child, someone who can't give informed consent due to their age, or someone who might have developmental delays and so is not necessarily able to understand and give legal consent. So they have assent, which means if they act or say that they don't want to be a part of it, then we take them out of the study, even if we have informed consent from a parent or guardian. It's an extra protection for people who are at higher risk. In the informed consent, you're also told you can leave the study at any time without penalty, and contact information if you have further questions, things like that. Confidentiality, so the right to concealment of identity on all information collected, written reports, informal discussions. You have the right to knowledge of the results in language appropriate to your level of understanding of what the study finds. You have the right to beneficial treatment. So if you're a participant in a control group, you have the right to alternative beneficial treatments if they're available. And then the question of deception. So with some studies, deception may be used. So where you're told untruths, you're deceived for whatever reason. Sometimes it's because there's concern about getting accurate data, or because like with medication trials, we can't tell people who's in the control group, right? That could potentially ruin the study. So when deception is used in a study, and yes, the review board can say it's okay to use it, afterwards you have to debrief your participants. So the investigator must debrief, provide a full account and justification of any activities when the research is over. And that is to try to build that trust again. So finally, our textbook has sections on the impact of COVID-19, and that is because it has significantly impacted development. So each chapter will address some of the impacts COVID has had on the age group. The example shown here, life expectancy or predicted number of years a person born in a particular time period can expect to live, has declined for the US population from 2019 to 2020, from 78.8 years to 77.8, so a drop of 1 year. And that it differed in terms of sex. So men, it declined 1.2 years, so from 75.1 years. For women, it declined only 0.9 years from 80.5. We also saw differences in life expectancy declines across different racial and ethnic groups, which is what the graph shows here. So you can see a decline in life expectancy for Hispanic, non-Hispanic whites, and non-Hispanic Blacks individuals. Reasons for these differences will be further explored throughout the course. Thank you.